Hello and welcome to Nodes 2020. I am Michael Black. I am a Neo4j Ninja. A Neo4j Ninja is a community member like yourself who's giving back. We help others get answers to their questions, and we also receive some exclusive perks and training. Ninja has to be a certified Neo4j developer. Once you become a certified Neo4j developer, you can become a Ninja too. Learn more at community.neo4j.com. Introducing our presenter, Matthew Sellwood, Manager at IQVIA. He will be talking about molecules or graphs, graph data science for drug discovery. Give a little background on Matthew. Matthew Sellwood, currently a product manager for IQVIA, will share an open source project that makes use of graph data science library for lead optimization of molecules in drug discovery. The project makes use of open source databases alongside the Graph Data Science Library to find new insights that could help chemists decide what molecule to make next in the process of designing a potential new drug. Matthew, I hand it off to you.
All right, thank you very much for that introduction. So as you said, my name is Matthew Sellwood. I'm currently a product manager at IQVIA. What our group does is we look for predicting different patient outcomes. But today, what I'm going to be talking to you about is a slightly different subject, which is close to healthcare. It's the bit before you get to the bit where you're taking the drug. It's the actual inventing of the drug itself. And this is something of a side project for me. But this is about chemistry and molecules, how they are actually graphs in themselves, and how you can use these in combination with some of the graph data science libraries that we've got to actually help in your drug discovery process. So why chemistry? So as I just said, I guess like molecules are actually graphs. So the nodes are the atoms, the bonds are the edges. And so here, this is caffeine. You've probably all had caffeine this morning when you woke up in your coffee or your tea. Here you can see the structure of caffeine represented as the graph structure. So here, as I said, we've got a nitrogen atom here. This is connected to a carbon atom. Carbon atoms are not normally drawn in chemistry structures. And this is a single bond, so a single type of edge, and then we have a carbon atom connected to an oxygen atom, and this is a different type of edge. So this is a double bond. So this can be applied as a label to an edge in a graph. You can say this is a double bond versus a single bond, and this is an aromatic bond here. So you have different types of edges, you have different types of nodes. Molecules lend themselves very well to graphs. One other way is actually you can represent the changing of different molecules and the forming of new molecules as graphs too. So the actual reactions that you form as part of the chemistry. And so when you're making caffeine or you're mixing reagents, just like basically baking, you're mixing, say here we've got node A or molecule A reacting with node B, molecule B. And here, these are forming, so these might be in your first representation of the graph. These are completely separate, they're not connected. But once we do the reaction, we form a connection, we add this bond in, we add this edge into the graph. And now we have our new graph where we've changed it to add in an edge. And you can actually look at these changes in a graph themselves, and we call these graphs reaction graphs. And I'll go into that in a bit more detail later. But generally, the generally thing to take away with this is actually, chemistry is one of probably the first things that used graph representation. It was one of the things that, graphs was made for chemistry. It's the perfect thing to use it for. Why should we care about chemistry? Well, I mean, hopefully I don't need to tell you, but everyone is made of molecules. So, all of the parts of you, your DNA, that's made up of different nucleotides all joined together. So it's all of you is made of molecules. And the things that kind of we need to look at here, there is a lot of different companies that are looking at molecules and how to put them together. So gas and oil companies. So here, the oil that you pull out the ground, the petrol that you put in your car, the plastics that are being made from this oil are all made from organic chemicals. So chemicals made from carbon atoms, and this forms the basis of a lot of our chemistry, including cosmetics and pharmaceuticals, and electronics. So there's a lot of different chemistry. So in your cosmetics, so the things that you maybe did your makeup, you did your lipstick, this is all made from different chemicals. In your electronics, so in your iPhone, in your PC, there is a lot of silicon. There is a lot of your batteries are made up of lithium ion, so they're made of lithium. You also have things like gold and really quite valuable metals on the kind of the circuit boards inside your electronic devices. So really, chemistry is at the root of everything that we do. But the thing we're going to talk about here is actually pharmaceuticals. So pharmaceuticals are very, very expensive to make, but very, very valuable. So these are things which really make a difference. So if we take the COVID-19 crisis at the moment, pharmaceutical companies are working out ways that they can put drugs onto the market, such as remdesivir, that can reduce the burden of the disease if you catch it. So these antiretroviral drugs that they're taking, that the president has just taken, really help to slow or help to prevent, in some cases, the disease getting worse. But drugs are useful. You take them all the time. You take them for a headache. So aspirin, for example, or paracetamol, Tylenol, as you might know it, is you take this all the time if you've got a headache. But the cost of producing every new drug is hugely expensive. So it's estimated to cost $2.6 billion to produce, on average, like a single new drug to market. And a significant chunk of that cost comes from actually, the research in developing the drugs. Often for every single molecule, every single drug which reaches the market, there will have been hundreds of thousands or tens of thousands of different molecules made to produce that one compound. It's very much a design, a test process. So it's a design, make, test process. So we have to make the molecules in order to test to see whether they work, in order to then produce, then iterate. And it's an iterative design process. And every time we do an iteration, it's expensive. So if we can reduce down the number of iterations that it's making to make this final molecule, then the more efficient we can be and the more money we can save the industry. And actually, the quicker we can get drugs to patients. And we can reduce the cost to patients when they're taking their drugs as well. So this is really, we want to get drugs to market quicker at lower cost. So the one thing about chemistry data is that it's huge. It's really, really big. This is big data. And chemistry, so I'm going to talk about three different areas of chemistry data here. So I mentioned reactions earlier. But structures, so that structure of caffeine that I showed earlier, this is a representation of the structure. And if we were to put together a database of structures of different molecules that are in just the drug-like chemical space, so molecules which have properties which are believed to be ideal for drugs, this space is estimated to be somewhere between 10 to the 23 and 10 to the 60. I mean, these are big numbers, and storing all this data is really, really difficult. In the background of this slide here, you might have noticed this very colorful bunch of dots, basically. This is a representation of 10 million molecules that are sampled across the GDB17 database, which is produced by Jean-Louis Raymond's group. And this is basically what they've done is they've enumerated. So they've basically enumerated in a computer every single molecule up to 17 heavy atoms. So that's every atom that isn't hydrogen, basically. So carbon, oxygen, nitrogen, things like that. They've enumerated this up to 17 heavy atoms. And this database is 166 billion molecules. And this is just a sample across that data of 10 million molecules. And you can see here, this to get an idea of the space. It's colored by the properties of the molecule. And so here we have, kind of properties that fit into this blue space. And then as we go down here, we kind of get different shaded, and eventually we get down to this red area here. And you kind of have these small clusters as well. You've got this other cluster here, which is maybe not quite as homogeneous as the rest of the space. But yeah, it's just really, really big. And kind of sampling across it is very difficult, and generating it is difficult as well. And the size of this, I guess, to give like GDB17, 166 billion molecules, to store this data in computational format compressed is around half a terabyte of data. So it's really, and that's a very small fraction of this 10 to the 60. So, okay, we've got all these structures of molecules, great. But actually, in order to design a drug, in order to design something that someone can take, you need to know the properties of the molecule. So the properties of the molecule are things such that, no one really wants an injection when they take a drug. Someone really wants to, we prefer to take our drugs orally. So we prefer to, take them with a glass of water. In order to take drugs orally, we need our drug molecules to be soluble, so that they actually dissolve in the fluids within our stomach. And not only do they need to be soluble, they also need to get from our stomach to where they need to be in our body. So imagine you have a kidney disorder and you need a drug to take to get to your kidneys. You need that drug to be able to go from your stomach via your bloodstream into your kidneys where it's needed. In order to do that, drugs need to permeate into blood cells. They need to get through the stomach lining. And to do this, they need to be very greasy. So they need to be both soluble and greasy. And if you've poured oil down the sink, I would advise doing that. The drain companies will have a nightmare. But basically the reason the drain companies don't like it is because oil and water doesn't mix. So here we're trying to find drugs which are in that perfect sweet spot of not too greasy, not too soluble, and we need to be able to measure these properties. And if we're going more advanced, we need to think about, okay, once it's got into the place that I need it, how do we make sure it's not toxic? How do we make sure it actually does what we want it? How do we make sure that it doesn't stay around in our bloodstream for a long period of time? We really need to measure a lot of different properties, and it's a really fine balancing act. But the issue with this is that because there are so many properties that we can measure of molecules, there's really kind of hundreds of properties. The data that is stored is not complete. So often and it's expensive to measure these properties too. So when pharmaceutical companies are producing drugs, they only produce the data that is needed in order for them to make the next decision. So this means that we have a very sparse data set. So we have maybe some molecules over here have got their solubility measured. Maybe some molecules over here have got their membrane permeability measured. Maybe some over here have got both. But a complete data set is very hard to come by because it's expensive to generate data on all these molecules. So what we need to do is make the most of the data we've got, use techniques like inference and predictions in order to learn from a space over here and see if it can be applied to a space over here. Making the most of this sparse data is what is really valuable in drug discovery, because it's just so expensive to be able to generate the data that you would need otherwise. And then I guess reactions I mentioned earlier. So when we're talking about how big data is, the largest reaction database contains 42 million reactions, or just over 42 million reactions. This is the Reaxys database, but this again is far from complete. So with these 42 million reactions, we might not even be able to make all of these molecules which are represented here. We might only actually be able to make the ones which are down here. So even though this seems like a big number, in chemistry world, it's still very small. So we have big data, but we always want more, and it's not nearly big enough. And we have to make do with what we've got. And graphs are very good at representing this data. So graphs are great at representing sparse data. In a table, you would have null values or nans, and you would have to store that data in the table to show that it wasn't there. So this property, it doesn't exist for this molecule. But in a graph, you just don't have the edge. You just don't have an edge that connects that property to to that molecule. You don't need to represent it, so you can actually store this data in a much more efficient way. Right, so now I've talked about data. Now we can actually go on and talk about, okay, how can we use inference and some of the techniques here. So in inference, so one way that we can do this is use a very simple technique called matched molecular pairs. This is a strong statistical technique, and it's based on single point changes in your molecule. This is known as also as graph mutation. So basically you're changing a single part of your molecule for another part. So here in molecule 1, everything is the same apart from this pink square. In molecule 2, everything is the same apart from this orange square. So we've changed this pink blob of our molecule for our orange blob. And you know, this is the sort of thing that chemists do when they're designing a drug. They'll say, okay, what happens when I change pink to orange? Does it make my molecule more soluble? Does it make it more likely to be distributed to the place that I want it? Does it make it more toxic? Does it make it less toxic? And these are the sorts of things that chemists are doing. They're making these changes, and we build up a database of these changes, and we can actually say, okay, imagine I now have a database of where I've changed pink to orange on a number of different cores. So if we call this the core, and we've changed pink to orange many times, we can then start to build up some data, and we can actually build a delta of, okay, statistically, when I change pink to orange, I get an increase in solubility by about 10%. And this data we can then store as an edge in our database. So we can say, when I go from fragment pink to fragment orange, then I get a change of 10% in solubility on average when I use that, no matter what core it is. And so what we've done is essentially use that data, stored it in a knowledge graph in a Neo4j database, and I'll go into it now a bit more about why we would do that, and kind of then a bit more about sorts of the different things you can get from that in terms of information. Okay, so matched molecular pair knowledge graph, what is it? So it's an open source Python package for building a Neo4j database of these matched molecular pairs that we just talked about. It's located on GitHub at the moment in my GitLab repo. It's still under quite a lot of development, so it's a fairly new repo, so it's not very polished at the moment. But the reason it was built is to develop and extract new insights from the sparse chemistry data using graph technology. And the number of different things that we use to build it are some existing open source toolkits. So the RD-Kit, this is a existing chemoinformatics toolkit written in Python and C, and has a lot of different libraries for handling chemistry. So this is built to handle chemistry. This is the library you want to use if you need to write chemistry in code. And then part of this library, there is a separate repo here, which is MMPDB. And this actually does the building and identifying of the matched molecular pairs from a given chemistry data set. And then once we've got the matched molecular pairs, then we ingest it into Neo4j. Right, so the schema. So this is the schema of the knowledge graph. So just talk you through it a bit. So I mentioned earlier about the molecules. So these orange nodes represent an entire molecule. So this is the entire structure of that pink orange structure that I showed you earlier. And here is another one. So this is molecule 1, this is molecule 2. This is just a small segment of the graph, just for showing. So there's many more molecules in the graph, as I'll show later. And each of these molecules can then be broken up into smaller molecules, which are called fragments. So this here is maybe our orange square. This here might be our orange triangle. This might be our pink triangle. And so here, what we're saying is that these molecules are made up of these smaller fragments of molecules. And these two molecules, as you can see, have got an edge. So these two molecules have got an edge saying that they are matched molecular pairs of each other. It goes both ways. because they are both matched molecular pairs is bidirectional. It doesn't have a bias about the direction. If you're matched molecular pair in one direction, you're also a matched molecular pair in the other. And then and the reason these two molecules are matched molecular pairs of each other is because they share one part of the molecule, aka these fragments, which change. So this molecule has this fragment, and this molecule has changed from this fragment to this fragment. And then the change and what it has produced and the property change is then represented on this edge. So in these cases, so these are all different environments. So this is represented here. We can really consider this as a single edge, but for technical reasons, chemists like to represent things as multiple different environments. So this is a kind of a technical limitation here, but basically we can represent this green thing as really one edge, but from more advanced use cases, we can represent it as multiple environments of how these fragments are encoded and how the property changes depending on how that fragment is represented with respect to the molecule. But basically, what we have here is we've changed this molecule for this one, and we have a property to say how much it's changed on the edge. You can also see that this molecule also has this fragment, but this fragment is not one which is similar, is not changed for anything that is similar for this one. This is just a different part of the molecule. So there's no data currently that represents this. Of course, these molecules have many more fragments as well that aren't represented here, but this is just for the illustration. So next we're going to talk about, okay, now we've got our data in our graph. And I should say that the data that we're using in this case is a drug discovery data set, one that was provided by AstraZeneca as part of their open source drug discovery initiative. Inside the Kemble. So these are properties that I was mentioning earlier, like solubility, binding of how much the molecule binds to your blood cells, how much it permeates into other things. So these are the properties which I've loaded into the knowledge graph. And what we're going to look at is 3 different graph data science algorithms. So the first one is node similarity. And what we're doing here is we're looking to see whether we can find compounds which are similar based on the fragments. So the parts of the molecules that you saw earlier where we're fragmenting it, what we're looking for is, okay, what percentage of those fragments are similar between other molecules? So this is a measure of similarity. There are other measures that are already well known in chemistry for similarity, and we're going to look at that, how we benchmark against those other methods. And the reason you might want to do this is because when you're designing a molecule, properties that are similar molecules which are similar often have similar properties. And so when you're designing a molecule, if you can say this one is similar to this one, a chemist might go, oh, maybe I'll make that one. That one might be interesting. The second algorithm we're going to look at is PageRank. This is to look at a slightly different property of the graph. So here we're actually looking to learn something not about what to make next, but here we're actually looking to look at what chemists actually do. So this is looking at, okay, what are the fragments which have a large impact on the network? And basically the hypothesis here is the fragments which have a large impact on the network are also the ones which a chemist would change the most frequently. So this is the sort of thing we want to look to find out. And then the final one is we're going to actually see, okay, can we actually predict new links between fragments? Can we actually suggest new changes that a chemist might make? The chemist should instead try this if he's not tried that already. So this is what we want to look at with link prediction. So I'm going to dive out into the demo now and talk you through some of the different use cases that we have that I just mentioned and go through them one by one. So this is set up in a Jupyter notebook here. So we've done this all in Python to show you today. So first of all, we're going to use the Python Neo driver to connect to our graph. Of course, we need a graph running. So in the background here, just to prove that I've got a graph running and it's real, we've got this our MMPKG database set up with a few properties that I've loaded in here, which contains 2,000 molecules and then 100,000 edges between them. So we can just open this up. and we can have a little play around. So let's just have a quick look at the compounds. So we can see here there's some compounds. These ones are matched molecular pairs of each other. If we double click on it, we'll see all the fragments of that compound. There we go. As well as some of the molecules that it's connected with as well. This is not very useful. It's just a big hairball. So really the better way to interrogate this at the moment is to use Python and pull out the data we want. So I'll just close that down and go back to our Jupyter notebook. So we're going to use PyTLEO to connect to our graph. We're going to use pandas to actually show the data. And here we have a whole load of imports from the RD kit. So this is so we can actually handle some of the data and actually visualize the chemistry molecules once we pull it out of the graph, because in the graph itself, the molecules are represented as strings, and we want to convert that into images so that we can actually show something. So, this is a developer conference, so, let's show some code. So first of all, let's run our imports. Let's connect to the graph. We've got the graph data science library installed within this graph, so let's just check that that's there. We need that for this to work. So yeah, so we've got our GDS, we're running 1.3. And now in GDS, the first thing that we're going to need to do is we're looking to create our in-memory graphs to run our algorithms on. So if you've heard some other talks throughout the day, an in-memory graph is created when you want to create a specific subset of your main graph in order to run your graph algorithm. So here, what we're going to do is we're going to create 3 or 4 different graphs. And this graph, the first one, we're going to call this the frag similarity graph, frag sim graph. And here we're going to pull in compound and fragment nodes, and we're going to pull in just the is fragment of edge. So this is just looking at compounds, the fragment, and their relationship about whether a compound is a fragment, a fragment is a fragment of a compound. So let's make this one. There we go. And then we get some metadata out, and it shows us how many nodes and how many edges got pulled out in this subgraph. Then we'll do another one. So this one, we're looking for a compound fragment degree to measure the degree of how many fragments are attached to each compound. I'll explain a bit more about why we want that later. And here we only want the reverse because we can calculate the degree in the other direction from this graph we've already calculated, but I'll explain a bit more about why we want the calculate the degree later on. Then here we want a fragment graph with all edges, and this one we're going to use for our PageRank algorithm. So that second algorithm we talked about. Here we've just got fragment nodes and then all of the edges related to fragments. We've also encoded a property on this in this graph as well, so we can use this to weight our PageRank. So this is the actual property of the data that we're looking at. So that solubility measure or that plasma protein binding measure. So like we want this in there so we can actually use it to weight our PageRank algorithm. And then the final graph we're going to make here is one that just looks at fragment edges, and this is to look at the degree between fragments. So we've got all our graphs created. Now let's check to see whether they're loaded into memory, and they're all there. So you can use this gdf.graph.list function to show the different graphs which are loaded. So if we run this, here we can see we've loaded our 4 graphs, and it gives all that metadata which is produced also in the JSON above. And just for kind of completeness here, if you do make a mistake, you can delete your graph from memory to prevent it using up using up all of the memory that you have. So this is just to show an example of how you would do that. You run this and then it produces the graph that it's dropped. Okay, so dipping into the first graph, so similarity. So find me compounds which are similar to others using fragments that they are made up of. So here we're going to call the node similarity algorithm. We're going to tell it to stream. So we want the data to be streamed back to us. And we're going to tell it to pull back the 2 nodes and then the similarity between them. And we're going to tell it to rank by most similar. And then we're going to convert it to pandas, and we're going to show them in the data frame. So let me run this. And then this takes a while. This is actually, most of the compute here doesn't actually happen here. It's this converting all the molecules to pictures, which takes a large number of time. So hopefully this will, there we go. Okay, so here we have compound 1 and compound 2. And to me, actually, these don't look massively similar. There's maybe one thing which is similar on this molecule. So this part here, this molecule also has it. But the rest of the molecule itself doesn't look very similar to me. These look quite different. Yeah, it's got a similarity of 1. So then we need to start to think about is this giving us what we need? Why are these molecules showing a similarity of 1? And one reason would be that in this case, when you fragment it, actually with the fragmentation algorithm that we've used to fragment the molecules, these molecules might only end up having a single fragment. And that single fragment is this bit. And in this case, those fragments are identical. So actually, maybe what we want to get a better idea of similarity is actually molecules which have a high number of fragments, and then look at the overlap between a high number of fragments. So, I mean, if we scroll down here, we can start to see molecules which are a bit more similar. But yeah, I mean, this doesn't seem satisfactory as it stands right now. So, okay, let's write that similarity data to the graph. So here, so we can use it later. So once we've got a bit more context around it. So, so we're going to use the .write part of the GDS library here to write these as new edges into the graph. There we go. Okay, so now we've written 10,000 new edges into our graph. So in order to work out, how many fragments a molecule has got, so we can actually divide it up, we're going to look at the degree. And so this is where the degree comes in. We want to actually look at, okay, where a compound has a large number of fragments and then compare the similarity of those fragments between the molecules. So we're going to calculate the number of nodes that are connected per fragment node, the number of fragments that are connected per compound node, and the number of fragments that are connected per fragment node. And now on each of these, and we're using the alpha algorithm here, degree. So if you read the talks, you know that these algorithms are in 3 main categories. You've got the alpha tier, which is kind of like the tier where you, it's like as kind of basic as it gets. You don't know whether it's been fully tested. You have the beta tier, which is where it's been moving into production, but isn't quite there. And then you've got the fully production tier of algorithms, which are like the most reliable, the most likely to scale to the data sets that you need. So this is in the lower one, but I've not found any issues with it so far. It seems to run pretty quick. So now that we've got our degree of our compound nodes and also other degrees written to the node itself, what we can say is, okay, now find me compounds where the fragment degree is greater than 15, aka compounds that have greater than 15 fragment nodes attached. It has a similarity score that's not null. And then we can actually say, okay, now return me the data ranking by that with our similarity score at the top. So here now, if I just go to this bit here, what I've done is here, we're doing our same thing as before. We're converting our molecules into our visualization. And then I've added 2 extra things on here. So this is so we can compare our fragment similarity to, the state of the art in chemistry. What is already out there for similarity? So here we're calculating Morgan fingerprints, and we're going to look at the Morgan similarity. These are also known as circular fingerprints. If you want to know more about these, then have a look at the RD kit and Google these. Then we also have max fingerprints. Max fingerprints are essentially fragment keys. So this one we might expect to be the most similar to our fragment similarity algorithm that we're generating here in the graph. So let's see how this works out. So here you can see we've just sorted by a similarity score again, and now we're getting something that's a bit more reasonable. Now we've looked at fragments that are there. These ones have 17 degrees between them. This is looking a lot more similar now. These look very similar to me. And then as we go over to our benchmarks of other scores, we can see that the Morgan similarity score is 0.5 and the max similarity score is 0.88. So the max similarity score is higher. So this is probably what we expect. It's more correlated. It's a more similar method Max has slightly more bits than we're encoding here. Here we're encoding 15 bits essentially when we're doing our similarity comparison. Whereas in the Morgan fingerprint, we're comparing 1024 bits. So this is much more granular. And actually, if you look at the literature, a Morgan similarity of greater than 0.3 at 1024 is considered to be similar. So actually, all of these algorithms are actually saying these molecules are similar, which is good. I would agree with them. So, and as you go down, we can see we've got more molecules. So this looks pretty good. We've got a similarity algorithm which kind of works. That's nice. Okay, so moving on. So next one is PageRank. So we're looking at ones which are important. So here we're going to use the PageRank algorithm here. We're going to write these straight to the graph. We're going to look at a weighted and an unweighted version, and we're going to compare these. So let's just have a quick look. So here we've got our PageRank, and we're sorting by this. And here what we're saying is basically, we were asking the question of what do chemists use the most? What do chemists change the most when they change a molecule? And they change hydrogen the most. This is pretty. This seems sensible to me. You know, when you're looking at a molecule, most of the atoms in a molecule are hydrogen. When you're doing organic chemistry, so it's highly likely that you're going to change hydrogen. The next one down here, we've got this benzene ring. So this is an aromatic ring. This is something which is frequently changed in molecules, and particularly in drug discovery, because aromatic rings are nice and flat. So they're very good at fitting into proteins. They're good at binding into, and also nice and greasy. So if you want to add some grease in, you tend to add in a nice aromatic ring, get some like a good balance of mix of properties to get just enough greasiness, but not too much. So aromatic rings are used quite a lot. So it's not surprising that we see aromatic rings cropping up here. And then we get different variations of this. So aromatic rings with nitrogen, bridged aromatic rings. These are all things that are coming up that are kind of changed quite frequently. So this again looks pretty sensible to me. We're learning some stuff about what the chemist is doing, and it seems from a chemistry perspective that these are producing some sensible results. Finally, we're going to look at link prediction, and actually one point to point out here is actually not useful to predict links between compounds here. So going back to our matched molecular pair algorithm, the algorithm that we have in place says that a compound is either a matched molecular pair or it isn't. So there's no reason to predict it because every edge that could have been added is already in the graph because it's just a heuristic. So instead, what is more useful is to predict whether fragments will be related, i.e. will changing this fragment for this other fragment give me a change in a property I am interested in. So we're going to use the Ademi-Gadar algorithm for this. So this is currently one of the link prediction algorithms within the GDS. And this is used to compute the closeness of nodes based on their shared neighbors. So basically this will tell us the likelihood of a relationship between two fragments. It's a bit like, I guess, like a recommendation engine, such as you get in Amazon. So here what we're doing is we're saying, okay, run the Ademi-Gadar algorithm. This one runs slightly differently. Here we're matching this pattern first, and then we're putting it through into Ademi-Gadar. So we're making sure we only get the fragments we want. We're making sure we have ones with high compound degrees. So we make sure we actually have, they actually have shared neighbors. There's no point comparing things that don't have shared neighbors or have any neighbors at all in that sense. So we need to make sure that they have neighbors. Here we've selected 10 or greater than 10 in this case. So what are we going to get back here? So here we get our score, and because we've not specified a direction, actually we get things in both directions. So the algorithm is not differentiating between, people often change this for this because they also might change this for this. It doesn't have the data to distinguish. So we actually do get both back in this case. So what we're saying here is we get this bridge carbon atom back for a swap for a simple methyl group. This is something which you might want to do if you wanted to look at changing the lipophilicity of a molecule. So this seems like a sensible change for me, particularly for this particular property of solubility. If you're balancing the actual lipophilicity and solubility against each other, then making this sort of change is sensible. Again, these changes here seem pretty sensible as well. We're going from an oxygen that's in some sort of ring system to one where it's just kind of branching and flapping around. Again, this is useful. You might want to go away from introduce rigidity into your system when you're wanting to try and bind a bit better to a particular protein or change the solubility. So these seem like fairly sensible changes. Again, going from kind of like non-aromatic fluorines to aromatic fluorines. Yeah, so I mean, these seem like some sensible things that you might want to try as a chemist. So we can go to our bench, we can try them out, we can see if they have the change in the property that we're interested in, we can see whether they do improve our solubility or not. I think one improvement here would be to actually add the directionality, because at the moment we are getting two ways round, which is a bit confusing, but yeah, I think they seem to be producing sensible suggestions, which is good. And then if we just scroll down past this, we have a little bonus here. So at the end, just wanted to look at, okay, given our similarity algorithms, can we actually plot them such that we look at how granular our similarity algorithms are? Does it give us the distribution that we're looking at when we're comparing to our max similarity or our Morgan similarity? And so if we look at the plot here, this is a bokeh plot. I quite like bokeh plots because you can add these tooltips, which is quite nice. So here we have our max similarity algorithm and our GDS node similarity algorithm. So as I mentioned earlier, max similarity it has a lot more fingerprints in. And so what we get is a much better distribution of similarity. Whereas on our node similarity algorithm, they're either kind of like not similar and clustered down here, or they're similar in here. So I think one thing which we could look at next is increasing that number from 15 to additional to a higher number, to see whether we can improve the granularity and get something that's more similar to max similarity. So that's kind of something which could be ongoing. So what we've shown you today is how we can use some of the graph data science libraries, such as the node similarity algorithm, the PageRank algorithm, and the link prediction on a matched molecular pair data set to infer new things, find new things out about the data that we're looking at, and try and make the most value out of the data. And I would encourage you to have a go with your own data sets if you're in pharma and you have these data sets, and try it out. I think there's a lot of value that can be added with these data sets, so I would encourage you to go for it. So finally, if you have any questions, I'll be free to take them now. Otherwise, you can feel free to contact me on either my email address. Either of my email addresses here. You can find the code here at this place. As I said, this is still ongoing in development. So if you want to contribute in any way, either to the code or also RDKit is an open source community as well. So if you want to contribute to the chemistry community, then please go to the RDKit code base. It's a real valuable use of Python code for chemistry, and see if you can contribute there. So yeah, thank you very much. And I guess just finally, there was, I think often one question that gets asked is where the data came from. So in this case, the data comes from Kemble. So Kemble is an open source database where different people have deposited and the Kemble institution itself has uploaded and curated a lot of data from scientific journals. So in this case, the data was, as I mentioned earlier, the AstraZeneca dataset, which was deposited into the Kemble database. So if you're looking for a chemistry dataset to get started on, then Kemble is a good place to start. It contains in kind of the low millions of molecules, and has a lot of different properties, and is a good place to start for chemistry data. Thank you very much for your time, and I look forward to any more questions that you have.